Are you able to see the number in the center of the circle? If not, you may be one of the 9% of men or 0.5% of women who, like myself, have red-green color blindness. Although not life-threatening, this condition can be debilitating and requires some adaptation in one's everyday life. To understand color blindness, one must first understand normal vision, or trichromacy. Trichromacy, also called the Young-Helmholtz theory, refers to the three types of photoreceptor cells found in the retina. These are called cone cells. Each of these has a different absorption spectra, absorbing photons and converting them to an electric signal sent via the optic nerve to the brain. Rods are another photoreceptor cell in the retina, and they contribute to color vision in dim light. Ospins, a group of light-sensitive proteins found in cones, are made up of a single polypeptide chain of hundreds of amino acids. This chain forms 7 hydrophobic regions across the membrane and is linked by extramembranal hydrophilic loops. It is believed that in any given cone, only one ospin gene is expressed, depending on the folding and looping of the chromosome. There are 3 types of ospins which each have specific photopigments to interpret light waves: short blue spectrum, medium green, and long red. Normally, all 3 function properly, but with color blindness, 1, 2, or all 3 are mildly to severely defective. Overall, about 75% of all color blindness occurs as a defect caused by green-sensitive cones. Anomalous trichromacy is the most common form of color blindness. All 3 cones function, however one is out of alignment, altering photopigment sensitivity. These 3 types are protanomaly, deuteranomaly, and tritanomaly. Dichromacy is the second type of color blindness with a complete absence of a functioning cone type, thus preventing perception of a specific wavelength segment. There are 3 types. The first is deuteranopia, which is the inability to perceive green spectrum wavelengths. Light blue is confused with lilac and pale pink with gray. Second, we have protanopia, which is the inability to perceive any red spectrum wavelengths. Black is confused with red or blue with purple. Finally, there is tritanopia, which is the inability to perceive any blue spectrum wavelengths. Here, it is common to confuse orange with red or light blue with gray. The final type of color blindness is monochromacy, whereby none of the cones work to absorb color. People see the world in shades of gray, very much like an old black and white TV. This is extremely rare, however, and only 1 in 33,000 people exhibits this disorder. Color blindness was first studied in 1798 by English chemist John Dalton. He and his brother both had a red-green type of color blindness, which led him to realize it was inherited. He mistakenly thought that the cause was discoloration of the aqueous humor, the liquid medium of the eyeball. It wasn't until 1911 that George Wilson asserted that it was a genetic disease of the X chromosome. Biologist George Wald received a Nobel Prize in 1967 for his biochemical discoveries in vision. He found that the 3 pigments of vision are made of vitamin A molecules linked to a protein. Each pigment is different due to that protein. His wife, Ruth Hubbard, carried it one step further by discovering that light changes the shape of the vitamin A molecule and triggers the vision process. The vitamin A molecule is called retinol. Because color blindness is most commonly inherited from mutations on the X chromosome and is recessive, men are more likely to inherit it than women. Thus, if a woman is colorblind, all her sons will be, but if only a father is colorblind, none of his sons will be, assuming the mother does not carry the recessive allele. It is important to note, however, that other causes can include macular degeneration, injury, medications, and aging. Obvious symptoms for color blindness include the inability to see shades or tones of colors, difficulty differentiating between colors, and rapid eye movement. Let me explain some of the several tests that have been developed to help diagnose the disease. The Ishihara test was developed in 1917 by Dr. Shinobu Ishihara and consists of 38 colored plates containing a background of dots with random numbers or patterns to determine who can see what colors. This is like the initial question that I asked you. 2 other famous color blindness tests were developed in the 1940s by Dean Farnsworth. These were a color hue test and a color arrangement test. Both rely on the placement of colors in the right order according to hue. Color blindness can at times be a handicap in one's everyday life. For example, Depending on its severity, reading charts and maps can be problematic. You cannot be a police officer, airline pilot, or a firefighter if you are colorblind, and in some countries you cannot even get a driver's license. Obviously, color blindness is particularly challenging for designers and artists where color choice is integral to the profession. Cooking can also be a challenge because people with color blindness have trouble telling when food is cooked through, or what is ripe, or what is green. To date, there is no cure for color blindness. However, it may only be a few years away. Researchers at the University of Washington have had success with genetic manipulation in squirrel monkeys and claim that a one-shot treatment on the retina for humans may be ready to test within the year. In the meantime, there are special glasses developed by the company Enchroma. These can be purchased for about $300 and claim to adjust vision using filters to correct for color blindness. It claims to work on about 80% of customers. In addition, there are apps available to adjust for color blindness, which can conversely simulate the disorder for those with normal vision. Unfortunately, the reviews haven't been all that great, saying that the apps don't always simulate the correct colors. In summary, color blindness is a genetic disorder that can vary from mild to severe and affect people's lives on an everyday basis. Although presently there is no true cure, scientists seem to be on the brink of a major breakthrough, offering relief to the ongoing challenges that it presents.